A multidisciplinary framework to characterize the physical properties of the bacterial genome, being presented by Xiaofeng Dai. Please.
It's really nice to kick off the afternoon speech. I'm Xiaofeng. I'm a first-year PhD student from the Betin lab. I'd like to take this opportunity to share with you how we use the super resolution technique to measure the physical properties of the bacterial genome. So my working system is E. coli. It's a flagship bacterium for the microbiology study. It has a genome composed by 4.7 million base pairs, which in length equals to 1.5 millimeter. But we know that the E. coli cell has a width of about 1 micron with a size of about several microns. So they compact this their genome into a membranous organelle called nucleoid, which is here. And this one, people used to think it as a randomer polymer meshwork, but it's not true because they have all these different DNA binding proteins to regulate this structure. And you have these codexin rings to actively change the local structure of the genome. Also, biological processes like transcription will introduce in supercalling into the DNA. So I just want to say, like, within this very dynamic structure, the physical property is an essential problem for microbiology and biophysics. It's because the DNA meshwork can control the accessibility of proteins of different sizes. For example, if you have a very condensed structure, it will exclude large size of proteins. And the viscosity of the environment will regulate the dynamic process of these diffusers. For example, if you have a very viscous region in here, and you will expect a slower diffusion for your enzyme. So my research goal is to measure the accessibility and the viscosity of the bacterial nucleoid under different conditions. To realize that goal, we use the passive microreology. By microreology, we specifically mean the viscoelastic property of the environment. And the passive microreology just means we don't introduce like the extrinsic perturbation to the system. To realize that, we introduce a probe into the environment of interest. And by performing the single particle tracking, we can study the dynamic of this probe. And this will enable us to infer the viscosity, the viscoelastic property of the medium. Specifically, in my research, I use a protein nanocage as my probe. It's a protein assembly developed by David Baker lab. It has a dodecahedron structure with a diameter of 25 nanometer. And it's very stable and it's extrinsic, so you don't need to worry about the binding events happening with the subcellular components. We fuse a green fluorescent protein to the probe and use an inducible promoter to control the expression level. And then we will see the diffusion of the nanocage under the fluorescence microscope. The real image looks like this, but if we want to have like more precision of our measurements, we want to achieve super resolution. We want to beat the diffraction limit. To realize that, we utilize this super resolution technique by taking the intensity profile from the detector and fit it with a 2D Gaussian. And the center of this fitting will be used as a localization for the probe within each frame. And by linking the localization frame by frame, we can have the trajectory. This enables us to study the dynamic of the probe. So now the nanometer scale resolution was achieved. And this similar framework has been utilized in the bacterial cytoplasm. And also, people use this strategy to study the genomic materials in the eukaryotic system. But the challenge for my system is the nucleoid is more crowded compared with the cytoplasm because it's a DNA meshwork, it's more condensed. And it doesn't have this physical boundary, this nucleus membrane in the eukaryotic system. So in this case, you know everything, every signal is from the region of interest. But in here, you don't know. It can be in anywhere. And to address that problem, we first have this a trajectory look like this. And within, for each cell, we determine the boundary by the segmentation algorithm. And we normalize the localization of the nanocage from each frame by taking the length of the long axis and the short axis. By having hundreds of thousands of localizations from thousands of cells, we can have this assembled 2D histogram to represent the localization pattern of the nanocage in a normalized cell. And this color here represents the localization probability of the nanocage at the specific region. You see a yellow color at the cell pole and a lower occurrence at the center. That means the nanocage is excluded by the nucleoid. So we can also use this exclusion and utilize this gradient analysis to determine the shape of the nucleoid from the heat map. And similarly, we can plot the spatial displacement map. The difference is like in this case, the color now represents the step size. So when you see a brighter color in here, that means the nanocage diffuse faster in a single frame. And we use a contour line to represent the nucleoid. By different methods, we see a very good colocalization for the nucleoid shape determination. So now we are very confident with this geometry of the cell and the nucleoid. But the problem is this is 2D imaging. So when you collect the data, you actually collect from the top view. And in real scenario, you will have the nanocage diffuse above and below the nucleoid from the side view. And they got projected to the same nucleoid region from the top view. So if you directly extract this dynamic process from this region, you have the portion. It seems like they are in the nucleoid, but they are not. So we need to address that problem. The strategy we use is to apply the 3D Brownian dynamic simulation. We first reconstruct the geometry of the cell and the nucleoid from this heat map. We assume a rotation symmetry along the long axis, and with this boundary, we do a rotation to get this nucleoid and the cell region boundary. And within this effective region, we can place random sampling the tracer in different regions, and we can also let it diffuse in this effective region. So now let's move to how we quantify the accessibility. We now have this reconstructed geometry, so we can randomly place the tracer in silico in the nucleoid region and also in the cytoplasmic region with different probability. And now the accessibility is defined as the density of the nanocage in nucleoid divided by the density of nanocage in cytoplasm. And by changing the parameter in the simulation, we can get this reconstructed simulated heat map. And we compare the simulated heat map with our experimental map. We use the L2 norm distance to quantify the similarity between the simulated one with the experimental map. And the one give us the lowest score. That tells us the accessibility of the genome in this specific condition. Additionally, we can also perform this dynamic simulation in this geometry, which means we set the accessibility of the nucleoid to 0, and we only let the tracer diffuse in the cytoplasmic region. In this way, we can reproduce the dynamic property of the tracer when they diffuse above and below the nucleoid, so that we can decompose the step size into 2 different regions. The goal is to have the displacement map from the simulation as similar as possible to this experimental map. Your eye may be drawn to the center part. You say, oh, it's not that similar. But the difference is because we set this accessibility to 0. So what we really should focus on is the decreasing, is the trend from the center to the edges. And this got captured from this simulation. And now we can extract the step size from the center region to get the dynamic property of the population when nanocage diffuse above and below the nucleoid. And that these step size distribution actually follow a Rayleigh distribution. And similarly, we can extract the step size from the experimental map to get this one. And now with the shape of the of population too, and also the weight can be extracted from the accessibility measurement. We can decompose this distribution into two populations, and the remaining portion is from the nanocage when they diffuse only in the nucleoid. Again, it fits with Rayleigh distribution, and by having the alpha, we can know the diffusion coefficient. And by Stokes-Einstein equation, the diffusion coefficient is inversely proportional to the eta, which is viscosity in here. And this enables us to quantify the viscosity of the genome for the first time. And we got a value in this case is about 180 centipoise. So you can imagine a 90% of glycerol mixed with 10% of water. So it's very viscous. And to remind you what we can do with this framework, we can use this static simulation to quantify the accessibility. And also, we can use this reconstructed structure to calculate the volume ratio of the nucleoid. It's another way to show how dense your chromosome is at specific conditions. We show that we can use the step size within the nucleoid region to get the nucleoid viscosity. But similarly, if we extract the step size within the cytoplasmic region, we can also get the cytoplasmic viscosity as well. So that's the 4 new dimensions, 4 new features we provided for this field, for microbiology, for like genome organization studies. Additionally, so we can further improve our technique by improving the resolution. We can even do the suborganelle resolution by masking the nucleoid into different regions. So to rationalize that, we separate this nucleoid into the periphery region versus the core region. And we see that the periphery of the nucleoid is more viscous than the core region, which is kind of counterintuitive because people will think like, you'll have a dense core and it's less accessible, so it must diffuse slower, which means a higher viscosity. And to rationalize that, we hypothesize this like a coupling between transcription and the translation may be the main reason for that. So to be more specific, in the bacterial system, in the prokaryotic system, it's different than the mammalian cell, than the eukaryotic system, because there's no physical boundary between your genome to the cytoplasm. So you can have the transcription and translation happen at the same time. And this gives you a result. When the RNA polymerase finds its binding site, it binds to the RNA, to the DNA, and it's transcribed mRNA. At the same time, the ribosome will find this mRNA and do translation, so you will have this polypeptide. At the end of the polypeptide, some of them, they will have a signal to find these anchors at the inner membrane. So in this case, you can imagine you have a linkage from the inner membrane to the periphery of your nucleoid. And with this, there's a tension applied to the surface of the nucleoid. That can be the reason why we have this spatial heterogeneity in here. How do we test that? We perturb the different biological processes. We use rifampicin to arrest the transcription. We use a chlorophenicol to change the translation inhibition, to inhibit the translation. We also use the amine and the pentanol to change the rigidity of this inner membrane here. We also thought about if the cell wall synthesis can be a reason for this spatial heterogeneity. And then we calculate the difference in viscosity by percentage in here. So if you compare to the control, we find the drastic change is from the rigidity of the inner membrane. So when you have a more rigid inner membrane introduced by the DDA, you will have more homogeneous environment in here. But if you soften this inner membrane, the change in viscosity actually becomes even larger. So these tests or hypotheses this difference in viscosity between the nuclear periphery and core is related to the coupling between transcription and translation in bacteria. So to summarize my methodology, we first used this single particle tracking to reveal the dynamic of the extrinsic probe for the passive microbiology. Then we use a 3D Brownian dynamic simulation to reconstruct a statistical ensemble of the 3D diffusion from a 2D tracking. We further improve our resolution to suborganelle. In this way, we can use the method to study how the biological processes influence the physical properties. For the sake of time, I only show you the application with the previous slide, but I actually have other results to show how each specific perturbation or biological process influence the genome viscosity and how environmental stresses change the response of the genome. If you're interested in that, I'm always happy to chat about that or just send me an email. So with all of these slides, I want to thank my lab, especially my PI, Professor Julie Batin, and our DPS team. We work all together to tackle the DNA, the chromosome organization, and our amazing collaborator from Indiana University. And thank you for your attention.
So normally when E. coli divide, symmetric chromosome puts a septum down, each cell gets a chromosome.
Yes.
There are certain strains that aberrantly put the septum down irregularly. So they create one cell which has 2 chromosomes and a mini cell which has none. And those are perfectly viable, well, whatever viable means in that case, but they respire, they're motile, you can look at them under a microscope, it's really cool. It would be really interesting to know how your probe behaves in that environment, because that's your perfect control. You're taking an aliquot of the cytoplasm, it's inside a membrane, it's got everything but DNA in it. That would be cool.
Yeah, that's very interesting. Yeah.
Yeah.
I also saw a work published in Molecular Cell, and they digest all the DNA, and they will have an empty cell with just a cytoplasm. That's exactly as what you mentioned.
Yeah.
And that's be a good control to see what's the pure cytoplasm viscosity and how different they are compared to the genome.
Yeah, and the other reason to bring that up is a long time ago, like 40 years ago, Pete von Hippel's lab at Oregon used those to show that most transcription factors or other DNA binding proteins in E. coli are always bound to the DNA. So finding their position is a 2-dimensional diffusion process and not a 3-dimensional diffusion process. So, you know, just because, well, we can talk about it later, but I just bring those up.
That's really great suggestions and questions. Thank you so much.